Continuing our talk on neurodevelopmental therapies, we have Dr. Mingshan Xu, who will be speaking on building a path towards genetic therapies for neurodevelopmental disorders. Dr. Mingshan Xu is an assistant professor at Baylor College of Medicine and the Caroline DeLuca Scholar in the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital in Houston, Texas. He received his PhD in neuroscience from Baylor College of Medicine and conducted postdoctoral research at the University of California, San Diego. The long-term goal of his lab is to understand the neural circuit dysfunctions in neurodevelopmental disorders and harness this knowledge to explore new therapeutic strategies for these disorders. For example, his lab created multiple mouse models of STX-BP1 encephalopathy and has developed a gene therapy for this neurodevelopmental disorder. His research has been recognized by several awards, including the Peter and Patricia Gruber International Research Award in Neuroscience, Janet Rosenberg Trubat Career Development Award, and McKnight Scholar Award. Please welcome Dr. Xu.
Okay. First, I want to thank Kit and Jill for the invitation. It's really my honor to speak here. My lab does not study TBRS, or at least not yet, but we are developing some ASO or AAV-based genetic therapy for other rare neurological diseases. So I hope that I'm able to share some of our experience and hopefully will be helpful to this community as well. So today I'm here really representing this group of talented former and current lab member, and just to present some of their work. As I said, my lab works on rare neurological diseases. So we are following a path going from the gene discovery of this neurological disease to developing therapies. So for example, once we discover the gene underlying these conditions, we usually build animal or cell-based models to study this disorder, and we use this model to understand the mechanism underlying those diseases. With this knowledge, we are able to start thinking about some of the therapeutic options. So this is where we perform the so-called proof of concept studies to test our idea. In our case, we focus on genetic therapies. If these are successful, we will enter into this so-called IND enabling studies, usually in collaboration with industry. So this is where we establish or validate these therapies, and if all of this goes well, then eventually you might get into the clinical study where for example, you perform natural history studies to identify some of the clinical endpoints for your therapies, and eventually you go through the phase 1, 2, 3 trials. And when all of these are successful, then you might actually have a therapy. So my lab really focuses on the area that from animal study to the IND enabling studies. So today as I said, my lab works on a few diseases. So some diseases we are still in the stage of building animal models. Some we have gone through sort of the pathogenesis study and start thinking about therapies. And there's one disease that we work on. We are actually in the IND enabling studies, and we are hoping this will get into a clinical trial in the next couple of years. So this is the one that I would like to share with you. So this whole research program was inspired by my encounter with Caroline, who had this disorder when I was still a postdoc. So when I started my lab, I decided to work on this disorder. So this disorder is called STX-BP1 encephalopathy, and it is one of the most frequent mutated genes in neurodevelopmental disorders. For example, these large-scale studies has sequenced about 30,000 patients with neurodevelopmental conditions, and on the x-axis is the name of the gene, on the y-axis is the number of patients carry mutation in a particular genes. So XTXBP1 turned out to be one of the top genes in this list. So this mutation in this gene also cause a variety of syndromes, for example, the Otohara syndrome, West syndrome, and even some rare case of Dravet syndromes. So it's one of the top pediatric epilepsy genes. As a scientist, one of the unique things about this disorder is that XTXBP1 pathogenic variants are highly penetrant, and if affect a variety of neurological functions, they are actually very common among the many neurological conditions. Therefore, we saw that if we can understand this disease, and perhaps it can provide insight into other disorder share similar features. So there, a study of about more than 500 patients of this disorder has shown that all of the patients have intellectual disability. Vast majority of them, over 90% of them, have seizures, and some are intractable seizures, drug-resistant seizures. And most of them also have motor deficits, manifest with dystonia, ataxia, and so on. And a subset of patients also exhibit psychiatric functions, such as hyperactivity, anxiety, or autistic features. So this disorder is caused by heterozygote de novo variant in the XTXBP1 genes. Because more than 50% of these variants are truncating variants, meaning they are deletion or stop codons. So therefore, we consider haploinsufficiency is the main mechanism. However, furthermore, there are also some missense mutation has been experimentally demonstrated that they destabilize the protein and cause a reduction of the protein levels. So this is the crystal structure of the protein, and you can see that the pathogenic variants are distributed throughout the protein. There are several recurrent mutations, but there isn't really a particular domain that's more critical for these proteins. So there's also some experimental evidence suggests that some of the missense mutation might have a dominant negative effect. But for today's talk, I'm going to focus on the haploinsufficiency case. So this gene encodes a protein called syntactin binding protein 1, or perhaps better known to the neuroscientist community as MUN18-1. It is one of the core components of the synaptic fusion machinery. It's required for synaptic transmission, because if you don't have these genes, then there's no neurotransmitter release, as shown in these studies. In the knockout neurons, there's no spontaneous synaptic vesicle fusion. So in the heterozygote states, which is what happens in patients, the synaptic transmission is reduced. This is a study from the heterozygote knockout human neurons, where you can see that the evoked synaptic transmission is reduced about half. This is the study from my lab showing that in the heterozygote knockout state, the GABAergic synaptic transmission is also reduced. So our research program started with the establishment of mouse model, and we characterized this mouse model, and we further understand what are the critical cell type for this disorder. So I will briefly go through this part of research and then get into the result of this therapeutic developments. So early on, we generated two null alleles in the mice. So both heterozygote of both alleles shows 50% reduction of the protein. So we performed a variety of neurobehavior study on these animal models, we observe a variety of neurological deficits. So here I list them but color-coded them in 4 different functional domains that's more or less correlate with the phenotype in human patients. And I will use some of these phenotype as an example to show you how we test our genetic therapies in these mouse models. So before we develop a genetic therapy, we thought we need to know what are the critical cell type for these disorders. For that, we created a flox allele, and we use 2 Cre lines. One target the GABAergic cells, the other one target the glutamatergic cells. When we created these animal models and we characterized their neurological phenotype, very interestingly, we found that the heterozygote mutation in GABAergic cells specifically recapitulate vast majority of the phenotype that we observe in the constitutive heterozygote knockouts. However, the glutamatergic cell also are important, particularly for one particular type of epilepsy phenotype. But the interesting finding is that there's very little overlap between these 2 mouse models, suggesting to us that both cell types are critical for this disorder. Therefore, if we were going to develop a therapy, we will want to target both cell types. So for us to start think with the mouse model, with the knowledge of cell type, the importance of the cell type, we start thinking about what can we do to develop a genetic-based therapy for this disorder. So there's critical question, as Harrison has mentioned earlier, which is about whether this disease or the neurological phenotype can be reversed or not, right? Because as a neurodevelopmental disorder, a question is always important is the damage permanent, or maybe perhaps the neurological phenotype is reversible. If it is reversible, is there a critical therapeutic window that we must intervene? Otherwise, it might still be too late. So we have developed a mouse model similar to what you have seen before. We can genetically reactivate the XDXBP1 gene in different stage, and I'm not showing you the result here, but the conclusion is that if we reactivate the gene in adult mutant animal, we can still reverse the neurological symptoms. So based on this, we thought, well, then a AAV-based gene replacement therapy will be ideal for this situation because we simply need to increase the protein level in the animals. So this is the approach that we have taken, and I'm going to show you what we have done for this. So to test this idea, we use a particular AAV capsid called PHP-EB, because if you inject this AAV intravenously, you can transduce a large fraction of neuron in the mouse brain. As you can see in an example here, we express a fluorescent protein, D tomato, and all of these yellow cells are basically transduced by this particular virus. So we confirm that we transduce both excitatory and inhibitory cells. In this picture, the NUN labels all neurons. The EYP is a genetic transgenic animal that expresses YP in all GABAergic interneurons. The PV and SST are the 2 interneuron types. From this image, you can see that clearly the virus has transduced both excitatory and inhibitory neurons. When we look at the protein levels, we saw that the virus was able to increase the protein level back to the wild type level. So in this case, where we have the wild type mice and the heterozygote knockout mice, the wild type mice also treated with the virus. So there's 4 groups. The wild type mice treated with the vehicle or virus, and the mutant mice treated with vehicle or AAV virus. As you can see, we were able to bring the protein back to the wild type level. This slide shows you the quantification. We included this wild type plus AAV group because it's important for us to find out whether or not whether too much protein is going to be a problem for this. So in the next several slides, you will see that each of these dots represent one mouse. The open circle represents female, the closed circle represents males. So first, this animal has the so-called hindline clasping phenotype, which is an indication of dystonia, perhaps. So here are the 4 pictures. As you can see, if you lift the mutant up, their hindline usually clasps together, right? So then if we treated this animal with the AAV gene therapy, several weeks later, this hindline clasping this phenotype essentially disappear. So here's a video to show you how they actually look like. I think it's quite obvious, right? So this mutant dramatically improved their functions in these particular assays. So we also perform other neurological tests, for example, the 2 motor assays. The first one is the foot slip. You basically put the animal on this wire grid and let them walk. So they have to precisely place their paw on the wire, otherwise they will slip. We basically measure how many times they slip as a measurement of their motor functions. Or you can put the animal on the top of lap stand and let them climb down this and get back to the cage, and we measure how long it takes for them to complete this task. So in the foot slip assay, you can see that the mutant animal treated with vehicle made a lot more mistakes than the wild types, but the treated or the animal received the gene therapy has dramatically improved their motor function. This is also true for the vertical pole test. The treated animal is essentially not much different from the wild type. Importantly, the wild type animal receive the AAV gene therapy do not show any phenotypes. So we also test their cognitive functions, right? In this experiment, we use the fear conditioning assay. So this is how this assay works. You basically place the animal in a chamber, and then you place a song, and then followed by an electrical foot shock. So this is a traumatic experience for this animal. And then next day, you put this animal back to the same cage, but without the song and without the foot shock. If this animal remember this experience from yesterday, it will freeze because they are scared. So we use this freezing behavior as an indication of their memory. Or you can put this animal in a different chamber, different environment, but play the same song that it heard yesterday. Then again, if the animal remember this experience, they will freeze. So we use this as an indication of their memories. So we measure this both contextual and the cue memory in our studies. As you can see here, the mutant animal clearly show a strong deficit in this fear conditioning or the fear memories, and both phenotypes were perfectly rescued by the AAV gene therapies. So lastly, I want to show you the epilepsy phenotypes. So for this, we performed the long-term video EEG recordings. So this is a video to show you that we have 3 EEG recording electrodes from the left frontal cortex, the right somatosensory cortex, or the left somatosensory cortex. The last channel is the EMGs from the neck muscles. And this vertical line indicates the current frame of these videos. So in the XTXBP1 heterozygote knockouts, they have frequent absence seizure like EEG signature, the so-called spike wave discharge. As you can see, this animal experienced lots of these spike wave discharges. So here is an animal, right? On the left, we have a video to show a mutant animal experienced the spike wave discharge. And this same animal, right, after the first recording, we dosed them with the AAV gene therapy. And then several weeks later, we record from the same mouse again, and you can see that the spike wave discharge essentially disappear from these animals. So here's the quantification. Again, as you can see, before the treatment, both the mutant have a lot of SWDs. The treated group dramatically reduced the frequency. So this animal model has another type of seizure called myoclonic seizure. And again, also this myoclonic seizure was complete rescued by the AAV gene therapies. So having gone through all of this, we are now entering the IND in labeling study in collaboration with gene therapy companies. So we are currently performing several important studies, including the pivotal pharmacology. For example, we are testing different doses of the AAVs to learn what will be the minimum dose that will be therapeutic beneficial. We also need to understand the distribution, the safety, the toxicity, and we are also working on the EEG biomarker and the molecular markers. So this last slide essentially summarizes our journey in this process. So we started this research. So the disease was discovered 2008, and we started this research program at Baylor in 2014. Over the years, we developed the mouse model and the gene therapies, and then about 3 years ago, essentially in the middle of the pandemic, we start collaborating with the company to initiate the IND in labeling studies. So right now we are at a phase where a natural history study is ongoing in the community. So hopefully this natural history study will last about a year or so. And so we are hoping that perhaps in 2025 we will be able to enter the phase 1 trials. So with that, I want to thank all the people that have done the work that I have shown you in the first slides. And we also benefit a lot from the core facility in our institute and the collaborators here. So this project has been supported by this funding source. Thank you.